Hello, this is Tommy. Welcome back to Chatomics. In today's video, I'm going to show you how to calculate partial correlation. And in the end, I will walk you through step by step a real bioinformatics example to calculate partial correlation of two genes CRISPR dependency score while controlling the cancer types. Okay, so what is a partial correlation? So partial correlation measures the relationship between two variables, for example, x and y, while controlling the effect of one of more other variables, for example, z. So what does it have to do with bioinformatics? For example, you are studying the relationship of two genes, gene A and B, and you observe a high correlation between their expression levels across many samples. But both genes might be regulated by the same transcription factor X. So is the correlation between gene A and gene B direct, or is it just because both are influenced by TF, this transcription factor X? So using partial correlation to test the relationship between A and B, controlling for transcription factor X helps you to answer this question. Again, it may be or may not be what you want. It really depends on the questions you are asking. So first of all, I'm going to show you a dummy example in calculating Pearson correlation, okay? So in R, it's very easy to calculate. Once you have two vectors, x and y, you can use this core function to calculate the correlation and use core.test to give you a p-value for the correlation. Okay, let's generate some of the dummy data. So, but before that, actually, I highly recommend you read this link here. Common statistical tests are linear models. So in this link, it shows the linear models underlying all those commonly used parametric and non-parametric tests. And correlation test is just one of them, okay? So let's load the libraries, set seed, and let's generate some random numbers here. And so this is the function to generate random normal distribution. And let's generate y, x. So y is just some random numbers, 50 of them. And x is another variable. So just 50 of them as well. Okay. And we can generate another variable y2. So this 2 times x plus some random. So that y2 and x, they are highly correlated because we generate the y2 using x. Okay. So let's add some new values here. So now we can generate this two data frame for example, this is a data frame of two columns, x and y, and you can use ggplot to look at their correlation. For example, use ggplot, and if you look at that, so you see x and y2, they are highly correlated because we generated y2 using x plus some randomness, so they're highly correlated. And x and y, they are actually not correlated. And if you look at that, it looks like this, okay? So let's calculate the correlation, just use the core.test, something that we are familiar with in R. So let's look at that. Core.test, and look at A. So you'll see the correlation is like minus 0.02, something like this one. And the p-value is 0.85, so they are not highly correlated, which intuitively look right. And we can just make them as a nice table, for example, like here. Actually, the core.test is equivalent to use this linear model in this, okay? LM is a linear model, and y is this one, and tilde 1 plus x. This is equal to this linear model. Y equals to beta 0 times y plus beta 1 times x. So essentially the null hypothesis will be, okay, whether beta 1 is 0 or not. If it's not 0, that means x and y, they are correlated, right? So we can calculate that using linear model, and then we can look at the TiDB. This actually is extract all those coefficients in a nice format from the Broom package here, which I didn't say actually from this package. So if we do that, let's see how it looks like. Okay, so this is the estimate, this is the slope, and this is the p-value, 0.856, right? It's the same actually as the core dot test, 0.856, okay? But then the correlation is different. So this is the correlation coefficient. This is the slope. This is the slope here. So they are not the same, but if you want them to be the same, so as I say here, the coefficient is not. It turns out we need to actually standardize x and y to get the same correlation coefficient, okay? So we are aiming to making this the same as the correlation coefficient. So let's redo this, but using the same linear model, but now we will scale y and scale x. Scale means you standardize x and y by minus the mean and divided by the standard deviation. Okay, so if you do that, and let's extract the coefficients from the linear model, beta 0 and beta 1, and look at the p-value. The same p-value again, but this time you have the correlation coefficient or the slope after you standardize x, y to be like minus 0.026, and this is the same as core dot test. Let's look at that core dot test. So this is the same as this, okay? So if you want to use linear model to calculate correlation, you need to actually standardize x and y by using the scale function to make the coefficient of the beta 1 equals to the correlation coefficient. Okay, so you can actually demonstrate that actually mathematically, so which I will skip. So beta equals to the correlation coefficient after you standardize x and y. Okay, now let's calculate the x and y1, which we show they are highly correlated. So again, we use the core dot test, the built-in function. Then we see the p-value is highly significant, 0, right? And then we have the correlation 0.88. If you don't remember, let's replot that. Actually, we already plot that. It's this one, x and y, to they are highly correlated. And okay, let's calculate the correlation coefficient using linear models. So remember, we need to scale y and x. And if we extract the coefficient, and the correlation coefficient is 0.887, it's the same as here using the core dot test, and the p-value is 0. So the p-value is smaller than 2.2. I think it's the minimal number that R can get. So okay, so essentially it's 0. Okay, so it's very satisfying to see that we get the same results using different methods, linear model versus the core dot test, the built-in R function. Okay, now let me walk you through a practical example using bioinformatics data. Okay, so that map, so this is actually from Broad. They have probably over 1,000 different cancer cell lines, and they profile those cancer cell lines using whole exome sequencing for mutations and RNA expression level, and also copy number, in this case, they also have the CRISPR screening data. So in that case, they actually knock out each gene for all the cell lines, and they measure whether that gene is essential for the cell growth. So that's a very powerful actually data set. People frequently go and use it to identify potential new targets for cancer therapy. Okay, so you go to the depth map. So there's a link here, and you go to here, then you have data download. So you have CRISPR screen, so you can download this data CSV file. So it's over 400 megabytes. So for that data, is a CSV file. Rows are models, or cell lines, columns are genes, and the values will be just how dependent that gene is for that cell line. Okay, and it also has other data: copy number, mutations, expressions, even have fusions. Okay. Okay, so I downloaded that CRISPR effect.csv file and saved that as a CRISPR score. So if you look at that after you read into R, and this looks like this: rows the model ID, or this is the depth map ID, this the cell line ID, columns are the genes. Okay. Actually, this is actually after I already cleaned that up. Let's redo it. So if I read that, okay, it takes a couple of seconds because 400 megabytes, it's pretty big. And you will find actually the first column, it's actually empty. So when you use read_CSV, the first column name will be ... .1. So we will see. So that's why it says, okay, this new name, this empty stuff becomes ... .1. So if you look at the first 5 rows and first 5 columns, it looks like this. So this is whatever the raw data if you read into R. And the first column name becomes this. And there's something strange here, like why you have gene symbol, you have parentheses, and then you have some numbers. So it turns out those are the untrans ID, and this within the parentheses, and we need to remove them. Okay, so we used dplyr rename the first column to be model ID, and this rename essentially all the column names. So remove like this, all these things like within the parentheses, and you can actually ask LLM or ChatGPT. So just tell them, okay, you have this stream and you want to remove them. It will actually show you how to do that. So that's a perfect usage of LLM. So if you do that and look at it again, now you have a clean data table. So as you can see, most of the time the data we downloaded, they're not necessarily what you want, and they're needed to be cleaned up, like the column names, or maybe there's some NAs that you need to fill in, for example. Okay. So we also need to read in the model.csv file. So you can download from here. There's model.csv. It contains the metadata information. For example, the cell line is from breast cancer or prostate cancer, different cancer types. So you have all this metadata information. So let's read in and look at that. So you have the model ID, which is a unique identifier for that cell line. And you also have the more commonly used cell line name, so people in the lab, they can recognize. Okay, but you also have this oncotree lineage, so know whether it's myeloid or it's a bowel, like colorectal cancer, for example. And if you look at that, use the table function, and you see those are the lineages that are included in the data sets. Okay, we can actually only select, we can create a new metadata, breast, and we mutate a new variable or new column here. So if the oncotree lineage is breast, we say it's yes, otherwise it's no. And in this case, we're going to calculate the correlation, it's CRISPR dependency correlation between two transcription factors, ESR1 or estrogen receptor, and also FOXA1, which is a pioneering factor and is known to be implicated in breast cancer and also ER function. Okay, the idea is that, okay, let's see whether ER and FOXA1 are codependent. Okay, so we have the CRISPR score, which is, take a look again. It's like this. And if you remember the metadata, let's take a look. There's 5 rows and 5 columns here. Oh. Breast meta, let me see. Oh, it doesn't have actually 5 columns, only 3 columns, because we select the model ID and the OncoTree lineage and then add another column for the breast, whether it's from breast cancer or not. Okay, and we essentially then will inner join the metadata with let's see, where's the CRISPR score? Okay, in this case, okay, now actually this probably I need to remove this. So we first inner join with the metadata. The whole metadata should with the CRISPR score, and they are joined by the model ID because it's the unique identifier. And based on that, we further actually mutate and add another column breast, the same like here. Okay. Okay. So if we look at that data frame, so in this case, we have the metadata information for the first few columns, but we also added all genes actually dependency score, for example, CRISPR. Like the gene names, like you have a lot of genes, like 20,000 genes almost, in the following columns after the metadata information. So what we can do now, we can plot. So first, always visualize the data using plot. So we can look at the dependency score of those two genes, FOXA1 and ER, estrogen receptor, and then we can color the points by whether it's a breast cancer cell line or not. So if we do this, and I'll split them by yes or no. And you can see here, you have the FOXA1 CRISPR dependency score here, and you have the y-axis of the ER estrogen receptor dependency score here. And if you split them by yes or no, it looks like in the breast cancer cell lines, they are more correlated. They are highly dependent and codependent with each other, which makes sense because when ER estrogen receptor plays a role in major role in breast cancer. Okay. Okay, so now let's actually calculate the correlation and the p-values, right? So we can, this is for the full data sets. So this is the CRISPR dependency score for FOXA1. This is for ESR1. If we do that for the full data sets, the correlation is 0.38. And if we only select breast cancer like this, because we have this new column called breast and equals to yes, and then we can calculate only the correlation from breast cancer cell lines. If we do that, you have a correlation of 0.51, which is usually much more correlated, and there's a very significant p-value here. We also have a very small p-value here when you use all the data. So here's the catch. When you have hundreds, thousands of data points, the p-value will be inherently small. Okay. In this case, pay attention to the effect size, which is the correlation coefficient here. Okay. But let's use linear models to calculate the correlation. So similar to the dummy example. And remember, we need to scale it for ESR1 and then scale it for FOXA1. And if you do that, use a summary, and actually we get this the same actually p-value. The correlation coefficient and the p-value is very small. And this for the full data set that you can see here. This is 0.737. Here, say we have 0.379, the same correlation coefficient. So we can now add the cancer type as a covariate to calculate the partial correlation. So in this case, we're controlling the lineage and still calculate the correlation to see whether it's still significant. So in this case, we just add the, this cancer type or lineage as a covariate. So this is still linear model here. This is your y, this is your x, and this is your x2, which is the lineage. And you do that. And by the way, because this is a categorical variable, and if you do this, look at that, it will use this adrenal gland as the baseline, as a reference, okay? So we can look at the summary of the partial correlation. As you can see here, still highly correlated, and now the partial correlation is a little bit smaller. It's 0.3, right? It was actually 0.37 if you don't use the partial correlation, right? So the interpretation is here, okay? So the FOXA1 coefficient is 0.3021. It means for every 1 standard deviation increase in FOXA1 dependency score, ESR1 or ER dependency score increased by 0.3021, okay? Controlling for cancer type. So it's highly significant. And let's see, as I said, FOXA1 and the ER have a moderate partial correlation, 0.3 here, across cancer types. This suggests that those two genes may be part of the related pathways, and indeed, they have synthetic lethal relationships, right? And within actually breast cancer, if we only restrict the cell lines here, the correlation is much higher, 0.53. And this makes logical sense given the importance of estrogen signaling in the breast cancer. Okay, so this is the final conclusions here. Without controlling, with controlling the cancer type is 0.3, and then if it's not controlled, it's 0.37. So it's a little bit inflated because it's primarily actually correlated in the breast cancer cell line, right? Okay. There are other usages of partial correlation. For example, when you calculate correlation, gene correlation to construct regulatory gene network. So you might want to use partial correlations. Okay, that's it for today, and thanks for watching it. And I will have the description, the R Markdown files in the description of this video, so you can follow along. And I have a blog post on this, you can also read it. So that's it for today. Happy learning. I will see you next time.